Dear students, today's lecture is Electrocardiography and Echocardiography in normal adults. Electrocardiography is a method of graphic recording of electric currents generated in the working heart. Contractions of the heart are preceded by its excitation, during which physicochemical properties of cell membranes change along with changes in the ionic composition of the intercellular and intracellular fluid, which is accompanied by generation of electric current. The most commonly used clinical ECG system, the 12-lead ECG, consists of the following 12 leads, which are bipolar, consist of 3 limb leads, first, second, and third, and unipolar, consist of 3 augmented leads, AVR, AVL, and AVF, and 6 chest leads, V1, V2, V3, V4, and V5, and V6. Bipolar limb leads consist of 2 electrodes placed at 2 different sites. These leads register the difference in potential between these 2 sites. The actual potential at either electrode is not known, and only the difference between them is recorded. Limb electrodes are right arm red, left arm yellow, left leg green, and right leg is black, is neutral. It means ground connection and not used for measurement. These leads form a triangle known as Einthoven triangle. Augmented unipolar limb leads, in contrast, measure the absolute electrical potential at one site. To do so requires a reference site, that is, a site at which the potential is deemed to be zero. The unipolar recording is then the potential sensed by a single electrode at one site, the recording or active or exploring electrode in relation to the designated zero or reference potential. The reference potential is the mean of the potential sensed by only 2 of the 3 limb electrodes. The electrode used for the exploring electrode is excluded from the reference electrode. The 6 unipolar precordial leads register the potential at each of the 6 designated torso sites in relation to a theoretical zero reference potential. And placement of precordial electrodes at first V1 placed on 4th intercostal space to the right of the sternum. 4th V2 placed on 4th intercostal space to the left of the sternum. V3 is on the 5th rib at left parasternal line, and V4 is at 5th intercostal space at midclavicular line. V5 is at the same place, 5th intercostal space, horizontal at the anterior axillary line, and V6 again at 5th intercostal space at the mid-axillary line. Bipolar limb leads, first, second, and third, form Einthoven's triangle. Other standard position for electrocardiographic leads are the augmented unipolar lead, AVR is right arm, AVL left arm, and AVF left leg. The lead axis of the 6 frontal plane leads can be overlaid to produce the Higgs-Axel reference system. Normal 12-lead ECG characterized by sinus rhythm. It means P wave is before QRS. Regularity, rate of is 60 to 90 beats per minute. Axis from 70 degrees to 30 degrees. Intervals. PR interval is 0.12 to 0.21 second. QRS complex less than 0.10 second. QTC is more than 0.46 second. Electrophysiological functions of heart. Heart characterized by automaticity, conduction, excitability, refractoriness, and contractibility. Automaticity is the function of pacemaker cells to produce spontaneously the action potential transient depolarization. Conduction, capability to impulse propagation through cardiac tissues. Excitability is the capability to become excited under the influence of impulses. These three characteristics can be evaluated by the ECG. And the next, functions of heart. Refractoriness is a property of cardiac cells that defines the period of recovery that cells require before they can be re-excited by a stimulus. Contractility is the capability of myocardium to contract in response to excitement. Conducting system of the heart. It begins from sinoatrial node from the 60 to 90 beats per minute, then spread to the atrioventricular node with rate of 40 to 50 beats per minute, then spread to bundle of his to rate of 10 to 30 beats per minute. Electrocardiograms is the record the electrical activity of the heart. It consists of the following waves, complexes, and intervals. The P wave is caused by contraction or depolarization of atria. The QRS complex is caused by contraction or depolarization of ventricles, larger than P wave due to more muscle. And the ST wave is caused during ventricular diastole or repolarization of ventricle. Checking of amplitude and time of ECG. For standard electrocardiography, amplitude is displayed on a scale of 1 mV to 10 mm vertical displacement and time as 200 milliseconds per centimeter on the horizontal scale. The normal ECG characterized by sinus rhythm, P wave before QRS complex, regularity, rate 60 to 90 beats per minute, axis from 70 to plus 30 degrees, intervals of PR 0.12 to 0.21 second, QRS complex more than 0.110 second, QT complex more than 0.046 second. Speed of ECG. There are two standard paper speed. There are two paper speed, 25 millimeter per second and 50 millimeter per second. 25 millimeter per second is the standard paper speed. It means 1 millimeter is small square equal to 0.04 second or 40 millisecond. 5 millimeter or large square equals to 0.2 second or 200 milliseconds. At a paper speed of 25 millimeter per second, one small square is 0.04 second, 5 small squares or one large square equal to 0.2 seconds, 5 large squares, 1, 2, 3, 4, 5 is equal to 1 second. ECG rhythm strip equals to 250 small squares or 50 large squares or 10 seconds. To calculate beats per minute, 1,500 small squares are equal to 300 large squares and 1 minute. Next paper speed is 50 millimeter per second. Why do we use 50 millimeter per second? We use for doubling the standard rate can reveal subtle ECG findings hidden at the slower rate. Here, this is the speed of 25 millimeter per second, and when we use the 50 millimeter per second, the hiddens can be easily found. Speed of ECG. If a different paper speed is used, calculation will have to be modified appropriately. Here it's written the measurements of the ECG. Detection of pacemaker. When the sinoatrial node is pacemaker, P wave occurs before QRS complex. In lead second, P wave is positive. Its shape is constant. Checking of heart rhythm. Compare R interval durations. In normal adults, the heart rhythm is regular. If you see here, the same. The variation of RR interval duration is less than 10%. Determination of heart rate. If the heart rhythm is regular, use the special equation. In normal adults, the heart rate is from 60 to 90 beats per minute. Detection of the mean QRS axis. Calculation of the mean electrical axis during the QRS complex from the areas under the QRS complex in leads first and 3. The concepts of the heart vector and the lead vector allow calculation of the mean electrical axis of the heart. Magnitudes of the areas of the two leads are plotted as vectors on the appropriate lead axis, and the mean QRS axis is the sum of these two vectors. And position of heart. It can be vertical heart position, horizontal heart position, and normal mean QRS axis. Normal heart axis and deviations. Normal can be between -30 and 90 degree. This is the normal heart axis. Left axis deviation between -30 to -90 degrees. Right axis deviation is between 90 degree to -180 degrees. And extreme axis deviation is between -90 degree to -180 degrees. Cardiac cycle. The cardiac cycle represents one complete heartbeat. It consists of the contraction of the atria, the contraction of the ventricles, and the relaxation of the entire heart. The electrocardiograph records the electrical activity that causes these events in the cardiac cycle. The ECG cycle is the graphic representation of the cardiac cycle. Elements of the ECG are waves P, Q, R, S, T, and rarely can be U wave. Segments, distance between two waves. Importance are P, Q, and ST segments. Intervals means wave or complex of waves with segments. Importance is PQ and QT segments. The reference levels for measuring ST segment deviation, depression of elevation is not the TP interval. The correct reference level is the PR segment. This level is also called baseline level or isoelectric level. Waves. The P wave characterized. These are P wave. P wave represents the electrical activity associated with the contraction of the atria or atrial depolarization. In sinus rhythm, there should be a P wave preceding each QRS complex. First P, then QRS complex. P, then QRS complex. Nota bene, the ECG cycle is recorded from the left to right, beginning from P wave. QRS complex. The QRS complex represents the electrical activity associated with the contraction of the ventricles or ventricular depolarization and consists of the Q wave, R wave, and the C wave. The ventricles are larger than atria and therefore require a stronger electrical stimulus to depolarize the ventricles. That's why the R wave is taller than P wave on the ECG graph cycle. Duration of QRS complex is 0.06 to 0.12 second. Amplitude is from 5 to 22 mm in limb leads and 8 to 25 mm in precordial leads. Q wave. In leads V1, V3, Q wave is absent. In leads V4, V6, Q wave is present. Duration is less than 0.04 second. Amplitude 1/4 amplitude of R wave in same lead. In lead AVR, Q wave may be deep and wide, or even QS complex may be present. R wave. Amplitude of R wave in V4 is maximal. Amplitudes RV4 more than RV5 and more than RV6. S wave. In leads V1, V4, amplitude of S wave is progressively decreased. Amplitude of S wave is maximal in lead V1 and S wave is of low amplitude of its absent in waves V5, V6. Amplitude of S waves more than or equal to 20 millimeters. The point during the transition at which the pattern changed from an RS to an RS configuration, that is the lead in which an isoelectric RS pattern is present, is known as the transition zone and normally occurs in leads V3 or V4. T and U waves. T wave represents the electrical recovery of the ventricles or ventricular repolarization. This wave. Note, electrical recovery known as atrial repolarization occurs following the P wave. This repolarization occurs at the same time as ventricular depolarization or QRS complex. Because of this, atrial repolarization is masked or hidden by the QRS complex and does not appear as a separate wave on the ECG cycle. U wave. Occasionally a U wave follows a T wave. It's a small wave that is associated in some as yet undefined way with repolarization of the Purkinje fibers or repolarization of the papillary muscles of the heart. PR or PQ interval. The PR interval is from the start of the P wave to start of the Q wave. It represents the time taken for electrical activity to move between the atria and ventricles. Duration of PQ interval is from 0.12 to 0.20 or 22 seconds. PR or PQ segment. It's on the baseline And the PR segment is the temporal bridge between atrial activation and ventricular activation. It's during this period that activation of the atrioventricular node, the bundle of His, the bundle branch, and the intraventricular specialized conduction system occurs. Atrial repolarization also occurs during this period. Marcus index is 1.1 to 1.6. ST segment ventricular recovery means onset on the ST segment in the junction or G point. The ST segment starts at the end of the S wave and finished at the start of the T wave. The ST segment is an isoelectric line that represents the time between depolarization and repolarization of the ventricles. It is contraction. In limb, leads ST segments is at the level of baseline or may be displaced less 1 mm upward or less 0.5 mm downward. In leads V1 to V3, ST segment may be displaced less 1 mm upward. In leads V4 to V6, ST segment may be displaced less 0.5 mm downwards. QT interval. It's equal to electrical systole. It includes the total duration of ventricular activation and recovery, and in a general sense corresponds to duration of the ventricular action potential. The duration of the QT interval decreases as heart rate increases. Rate. How to measure rate? Formula 1. 300 divided by number of big squares between RR waves. Formula 2. 1500 divided by number of small squares between RR waves. For irregular heart rates, get the number of QRS complexes in a 6-second strip, then multiply by 10. Rhythm. We should check the rhythm and answer the question, is there a P wave? Are the P wave same, regular or irregular? Check relation of P wave to QRS complex. Check PR interval, normally should be 0.12 to 0.20 second, and use the formula, this formula. This formula for relating QT interval duration to heart rate was developed by Bassett in 1920. The result is computation of a corrected QT interval or QTs by using the equation. The normal QT C is from 0.35 to 0.46 second in male and from 0.35 to 0.47 second in female. TP segment equal to electrical diastole. The TP segment, which begins at the end of the T wave of one cardiac cycle and ends with onset of the P wave of the next cycle is usually the most appropriate interval ECG baseline. And next instrumental method of heart is echocardiography. An echocardiogram, cardiac echo, or simply an echo is an ultrasound of the heart. Echocardiography uses standard two-dimensional, three-dimensional, or Doppler ultrasound to create images of the heart. Transthoracic and echocardiogram is a standard echocardiogram is also known as transthoracic echocardiogram or cardiac ultrasound. In this case, the echocardiography transducer or probe is placed on the chest wall or thorax of the subject, and images are taken through the chest wall. This is a non-invasive, highly accurate, and quick assessment of the overall health of the heart. Types of echocardiography are transthoracic echocardiogram, transesophageal echocardiogram, stress echocardiography, intracardiac echocardiography, intravascular ultrasound, strain rate imaging, three-dimensional echocardiography, contrast echocardiography. This is the norms of dimensions of the heart. Thank you for attention.